Hello? Hello? Hello?
Hello, good morning, sir.
Good morning. I was ready. I was just waiting.
Yes, sir. Yes, sir. Shall we start now, sir?
Please, please.
Yes, yes. Thank you, sir. So now let's start. I'll just check whether Professor Mathur is also here, sir.
I can see him. Yes, I can see him here.
Professor Mathur is there with me. Good morning, sir.
I have unmuted myself. Okay.
Yes, yes.
Hello, good morning, sir. Good morning.
Yes.
Mr. Mathur is from that school who will never be late. He'll be there 5 minutes before the event.
I will go on this 10 minutes, sir.
Very good, sir. And there was a problem in the entry, so that's why I just wanted to check.
No, please carry on.
Yes, thank you so much. So good morning, everybody. I have a great pleasure in welcoming you all to the week-long international webinar on COVID-19, Covinar, organized by the Department of Biochemistry and Molecular Biology, Pondicherry University. We have a panel of experts who will be enriching our knowledge on the various aspects of COVID-19, like structure and biology of coronavirus, immunological modifications in our body during COVID-19, simple interventions to combat corona infections, immunotherapies against COVID-19, and lessons learned from the pandemic. I thank all of you for participating in this webinar, and I'm sure you'll be greatly benefited by these expert lectures. Now, let's start the session with the university anthem. Now, I request our respected Professor P.P. Mathur, Head Department of Biochemistry and Molecular Biology and Dean School of Life Sciences. I should also say the driving force behind this webinar to deliver the welcome address.
I have to unmute, yeah. Honorable Vice Chancellor, Professor Gurdeep Singh, Dr. Rukmini, our resource persons, and I can see Dr. Pavithra here, our colleagues, faculty members, students, and outside participants. In fact, we have been seeing these kinds of developments very often now that most of the things are discussed in this mode now. Our department had already taken a lead in this way, and we had a very, very successful webinar, and the speaker was from Italy, and that was very well received. I think this will be surpassing that that kind of thing, because here I was told yesterday more than 1,000 participants have registered already, and many of them, they are just seeing the live streaming. They will not be able to participate in the discussions. But I think Professor Gurmeet Singh, Vice Chancellor of this university, he has been a real driving force to everybody, all the departments, many of the departments have conducted it. But this meeting is much different than others. First of all, the word which is coined by Dr. Covinar is a completely new thing. It is the first time I'm hearing it in terms of COVID-19. And it's very, very nice because all the discussions which will be centered around will be the COVID-19. So this is a new thing, and I'm sure that many people will follow this lead of naming anything which is related to COVID-19 and specific seminar as Covinar. I'm so glad that so large participation is there today. And in fact, I had already been telling her and others also to organize something or other. In fact, most of the people, because we are not used to arranging such activities in a very different manner, they took some time in organizing this thing. So, but anyway, it is very nice that she has organized this particular meeting, and the participants are across the globe. Even the speakers are from all over the country, and you would see that from abroad also participants. And the topics which have been chosen, and the topics are very varied and very, very timely, right from the molecular biology to the effect of COVID-19 in different our life, and also the kind of diagnostics which will come out. And now everybody has changed the meaning of good news. Is not the earlier way what we used to consider good news. It is like, if anything is there now for vaccine of COVID-19. That has become good news now. So I think that very soon some of the developments which are being discussed on this manner would be reality now. So I wish this discussions, and many of our students are present today here. They would try to participate in the discussions with the speakers, those who have come, and among themselves also. And I feel that at least half an hour when should be left for discussion, minimum half an hour when you have such a large participation. And you can record the whole lectures and can share it later on university website. You can share it on the university website. And not only that, these questions can be those who post the questions and the speakers may not have time to answer at this time, they can post the answers, and that also we can host it. So I think this would be a great seminar, and I feel that this Covinar which Dr. Rukmini has proposed and to be very successful, and it's a 5-day. I also told her that nobody has time and nobody has patience to listen for more than 1.5 hours. They have other things also, and the whole day they are doing like this only. So that's why she organized in such a way, and I am happy. And I thank our Honorable Vice Chancellor that he made his presence. Though I just sent a mail last night to him requesting him to join us, and he is here. So with these words, I wish this particular webinar a great success, and the learning exercise for most of the people, those who are participating in that. Thank you so much, and wish you all the best.
Thank you very much, sir. Now I request our Honorable Vice Chancellor, Professor Gurmeet Singh, who is the pillar of support for all the good things happening in our university, to deliver the inaugural address.
Good morning, everybody. Our Dean, Professor Mathur, Dr. Rukmini, who has taken the lead to organize this, Dr. Sudharani and Dr. Subha Lakshmi, the team which has been supporting this webinar. I'm glad you have given a new name to this called Covinar, because in the time of COVID, whenever you say Covinar, it will become more catchy and people will be more attracted to this, particularly the youngsters. And it has been very aptly entitled as COVID awareness during this seminar. This is on COVID awareness, which is very important, because at a time when the entire human race is feeling very depressed, passing through a very bad phase, to organize something like this, which will kindle a spark in the mind of youngsters that no lot of things can be done, even during these times when people are not feeling very encouraged, very motivated. A webinar of this kind would definitely encourage them to utilize their time better. I don't know, very many of you may not be aware of this, that there are many hospitals who are carrying out testing here in our city, Pondicherry, and they approached us if some of the equipment that we have in our biosciences departments can be used for this. We readily agreed for this. So in our own manner, in our own methods, we are helping the medical system here in carrying out the testing in a much better manner. And this is what is the need of the hour. And all bioscience faculty, if they can wake up to the existing difficult time, I think it will go a long way. And a webinar of this kind, which will create awareness amongst our faculty, amongst our teaching faculty, amongst all those who matter, and particularly the young researchers who are here, who have to initiate themselves into various research careers. I think they will be the ones who will be the maximum beneficiary of this particular one. They will get a direction that in times of this, what are the areas they can probably work on. And once you interact with some of the best experts in the area, I'm told there are 1,000 registrations which have been there for this particular webinar, and there are experts from all over. This will definitely motivate our youngsters to pick up the threads. I can see the names of Dr. Pavithra, Venkatagopalan, Dr. Shishu Paltra, and Dr. Vichumani, Dr. Gopal, Dr. Vijay Singh from the United States, and many others who are here. Dr. Muthumani is also from US, Philadelphia, and many more will be there who are participating. I would urge all our young friends, youngsters, researchers who are here attending this, that during the question hour sessions, they should feel free to put their doubts to these experts, so that I'm particularly keen on this, that they pick up the right kind of an area to start their research on. This is the most important factor in a researcher's career. If they are able to pick up the right problem, then they never look back. And if they get into a problem which doesn't take them anywhere, that leads to a lot of depression. As it is, the time through which we are passing at the moment is not a very encouraging one, as I said. And therefore, for them, for the youngsters, I think it is very important to cling on to a right problem at the very beginning of their career. I compliment the Biochemistry department. I compliment the entire life sciences faculty of our university, that in such a difficult situation, they have come out with the idea of organizing this webinar, and rightly coined a word called Covinar. I feel that at the end of this, they will. I would in fact suggest that they document this, they record this entire proceeding, and keep it in the form of a document, because even if 30% out of this, we can use it for the benefit of youngsters and for the benefit of society at large, I think it will go a long way. And it's a right initiative at the very right time. So therefore, my compliment, and I encourage, I urge all our youngsters to participate very seriously, put their doubts to the experts, interact with them, and make the best use of this particular webinar. I give my compliments to the entire department led by Professor Mathur for taking this wonderful initiative. And I not only hope, I am 100% certain in my mind that this will be of a great use to everybody who is attending this. I wish the webinar very great success, and once again, compliment everybody who has helped Professor Mathur in organizing this particular one. And the best part is that in such a short span of time, you have been able to muster over 1,000 people here for this. That proves, speaks in volumes, in fact, about the seriousness with which you have gone in for this. So wonderful work done. carry on such a good work in future as well and continue to organize more such webinars in future also. That's not the end of the story, it is the beginning, and the world is not coming to an end, let me tell you, with the vaccine that we are looking forward to. You have a long way to go. You have to continue to put in your best so that a pandemic of this kind never ever happens on this earth. It has led to the standstill of everything. So I hope with the hard work of our biochemists and biologists, life sciences experts, they'll be able to come out with something wonderful for the benefit of humanity. Thank you so very much for inviting me, and I wish the webinar a grand success. Thank you so very much. All the best.
Thank you, sir, so much.
Thank you very much, sir, for your kind words and encouragement. I assure you that we will join hands in the fight against coronavirus. Thank you. Now let's start the scientific session. Today we have with us an expert researcher in the field of microbiology, Dr. Pavithra Venkatagopalan. Dr. Pavithra Venkatagopalan is the director of Care Health Diagnostics in Chennai. She did her engineering from Anna University and her doctoral research from Arizona State University, USC. Prior to this position, she was teaching in Arizona State University. She has very good publications and several presentations to her credit. She has received several awards, including Excellence in Teaching Award from Arizona State University. She has done her PhD research on coronavirus, and hence, after the start of this pandemic, she has become the most sought after expert grower in the world. I'm sure most of us would have seen her in popular magazines as well as media. We are very fortunate to have her here with us today, and I now request her to deliver her lecture. Pavithra ma'am, please. Ma'am, I request you to kindly unmute your mic. Thank you.
Good morning, everyone. Thank you, Dr. Rukmini, for the very, very kind introduction. It's an honor to be a part of Pondicherry University. Thank you, VC Gurmeet Singh and Professor P.T. Mathur for inviting me to be a part of this. This is one of the biggest webinars that I have been part of, and it's making me a little bit nervous. But I think we'll all enjoy this journey of understanding the basics of coronavirus. Now, I'm going to share my screen and take you through the presentation. So I generally tend to talk a little fast. So if you have any questions that we don't get to at the end of today's session, by all means, please message me or email me in the contact information that I have given in the chat windows. I will be very glad to answer each and every one of the questions that are being addressed to me. Okay, so in order to understand the virus, there is a lot of misinformation that's going on about this virus. And frankly, when I graduated in 2012, I wasn't looking for a career in basic research, but that has a lot of different reasons why we choose the path that we choose to go. However, when this infection came out, me, like all of you, was receiving a whole lot of forwards that were completely meaningless and unscientific. And that got me to start talking to smaller groups of people about what this virus is and what this virus does. I come to recognize over time that even people that are in the field have many misconceptions about the virus. And I thought the best way to start understanding the virus is to know about the molecular biology and the life cycle of the virus itself. So this is where we will start. Since I'm not sure about the background of everybody, I'm starting a little bit on the basic side. So if I'm boring you, please bear with me. I promise I will get to the interesting parts. Now, what are the domains of life? When life evolved, we first had bacteria, which is a single-celled organism. Most of us are familiar with them as being pathogenic, but there are actually a lot more which are useful and not pathogenic. You have archaea, which are specialized unicellular organisms, but they grow under very specific conditions. And then we have eukarya, which cover everything from fungi and yeast, which make our bread and beer, to you and me. That is the rest of life. However, I have not mentioned viruses here. Viruses are very interesting and unique in the sense that they occupy their own position in the domains of, outside of the domains of life. So viruses are defined as obligate intracellular parasite. What this means is viruses are dead outside the cell. However, when they are inside the cell, they are alive. So obligate intracellular parasites. They absolutely need a host cell to function. This is what makes this group or this family of organisms most interesting to study, because it basically questions out the existence of what life is as we define it. Now, the second interesting aspect is we have the central dogma of life, where you have all the cells and organisms that we know in the domains of life have DNA and then RNA and then protein. So basically, if we want to see what DNA is, let me compare. Let's say you want to make a building. You go to the architect, you tell the architect what you want, exactly how many rooms, whatever, everything you want. One fine day, a week or so later, the architect will give you a nice little image that says, this is what your house will look like, this is how many trees you will have, this is the car parking that you will have. Now, this is what the DNA is. It contains a lot of information. But is this information useful? Now, can a contractor build from it? Not really. So you have that is being converted into drawings like electrical drawing, plumbing drawing, structural drawing, and things like that. And that is what your RNA is, which has the technical details specific for each and every aspect. So you have readable information that can be further translated. Then when the contractor looks at the technical drawing, which is what the cell looks at the mRNA, it makes the building blocks of functional cells using different proteins as dictated by the mRNA. So this is the other central dogma of life. Coronaviruses are very interesting, and as we know, biology is the science of exception. Coronaviruses in their life cycle do not have a DNA phase at all. They only have an RNA, and they have the mechanism, and they have the ability to make proteins just with whatever machinery that they have. Today we will be understanding these parts of the machinery so that you can build on what is going to happen in the future comes. Now, viruses, especially mammalian viruses, are broadly classified based on their genome. Viruses are either DNA viruses or RNA viruses. And RNA viruses can be in multiple forms. You have a positive sense RNA, that is a single-stranded positive sense RNA. You have a single-stranded negative sense RNA. You have both, or you can have something like HIV, which has an RNA but goes through a DNA stage in its life cycle. Here we are looking at a single-stranded positive sense RNA enveloped virus, which is what coronaviruses are. Now, coronaviruses belong to the order Nidoviralis, and they have the three families Coronaviridae, Archeriviridae, and Roniviridae. All three are generally known for infecting animals, not so much humans. And all of them have, especially Archeriviridae and Roniviridae, infect a lot of aquatic animals. We are looking at the family Coronaviridae, and it comprises three groups: Archerivirus, Coronavirus, and Torovirus. And we are going to be focusing on the coronavirus family of viruses. Now, coronaviruses are not new at all. A lot of people have heard the term coronavirus only now in the news. So they find it very interesting when I say coronaviruses are absolutely not new, and they have been known since the 1960s. And we always knew that they were causing minor human infections like the common cold, but they were economically extremely important for people in the farming industry, because if you have a chicken with an infectious bronchitis virus, IBV for example, you have a chicken farm, if you have the IBV infection, it can wipe out the entire farm. And if you have a cat, especially an infant cat, and if it gets feline coronavirus or a canine coronavirus for a dog, it is considered lethal. So a lot of effort was put into understanding coronaviruses from the perspective of a veterinarian. Humans, we always knew they infected, but until SARS coronavirus came in 2002, nobody really thought it was going to be a big deal. So from this, and they're also known to cause a variety of symptoms. So one of the questions that people always ask me is, what is this? When COVID came, they said it was a cold. Now they're saying there is blood clotting. Now they're saying there is diarrhea. We actually know that coronaviruses cause a variety of respiratory, enteric, liver, and neurologic infections depending on the virus. So again, none of these are surprise to the people that are actively involved in the field. So now, what are the notable coronavirus infections? Have we been infected with the coronavirus before? In all likelihood, yes. 30% of all common colds are caused by coronaviruses, which means if you get 3 colds in a year, one is probably caused by a coronavirus. So where did they come from? Most of coronaviruses are generally classified as zoonotic viruses, meaning they have an animal origin and they get transmitted to humans. With the growing population, and these are not the only zoonotic viruses. We know HIV was a zoonotic virus that jumped from macaques to humans. We know that Nipah virus jumped from bats to humans, which happened recently in India. Now, so animal to human transmission is not at all something that is new or novel. This doesn't mean we should be going about destroying all the animal habitats around us. This is the price we pay for living in a very crowded environment as the world itself is progressing towards. Most of the time, rabies is a zoonotic virus. Most of the time, the carriers tend to be bats. We don't know why this happens, but bats seem to be carrying a lot of viruses that can jump species to humans, but without having any detrimental effect on the bat colonies themselves. So this makes them excellent carriers because they carry the virus replicates well, and then let's say you have a big animal farm, and then you have sheds, and anywhere you know that bats are going to be roosting on the nests, roosting on the roofs of these farms. And when these bats make the dropping, and the people that go clean the sheds, they are constantly exposed to different types of viruses. I'm not saying they only got a coronavirus, they probably got multiple viruses. And the people and the viruses that survive well in the human mold continue to replicate. So we know the various coronaviruses, NL63, 2290, OC43, HKU, all these viruses, they've come in Netherlands, England, China, Hong Kong, and SARS came out from China. They came from these animals to either directly to humans or through an intermediate force. For example, 229E came through llamas. OC43 came from rats to cows to humans. SARS coronavirus, the one that came in 2002, came from bats to civet cats to humans. MERS coronavirus, which is one of the most deadliest forms of coronavirus that we know today, it still exists in pockets of very few number of cases that happens, came from bats to camels to humans. And now we have SARS coronavirus 2, which came from bats to humans. We just don't know what the intermediate force is. How did we identify that it came from bats to humans? One of the favorite conspiracies for everybody is the virus came from a lab in China and it is lab manufactured, and there is evidence for all these things. However, that is absolutely not the case because Wuhan Virology Institute is one of the most renowned virology institutes in the world, especially to study the zoonotic transmissions, because the area is an epicenter for various infection transmissions. So if you don't have such an expert in place like that, where they do a lot of serosurveillance and patient studies, we would not have been even able to identify the cause of this new infection, which has now become a pandemic. So when we look at the genomic analysis of this SARS coronavirus 2, it was identified to belong to the beta coronavirus group of family of viruses, which is very similar to SARS coronavirus and MERS coronavirus. And then when we look, initial sequencing was done of the first clade of viruses. It was found to be extremely close to these bat sequence, coronavirus sequences that have previously been known and characterized and archived. So that's why we're quite confident that the virus jumped from bats to humans. Now, the question everybody asks is, how do you know they did not manufacture it in a lab? First of all, if we were manufacturing it in a lab, we would have chosen better amino acid modifications for the virus to infect even better. Nobody, I would like to believe, is that mean and nasty and cruel to be involving themselves in biological warfare like this. Plus, there are, of course, more deadly viruses in the world than SARS coronavirus too. Now, when you look at what the virus looks like, you've probably seen an image like this on the left quite commonly in all news medium and publications. So the virus is enveloped, as you can see that there is a double layered lipid membrane, and then you have these large club-like projections on the viral envelope. These are called spike proteins, and this spike protein is what initiates the viral infection. Then you have the membrane protein, which makes up the backbone of the viral envelope, and then you have these smaller proteins in the viral envelope called E protein. We'll talk about what each of these proteins do in detail as we go further. Inside the viral envelope, you have that single-stranded positive sense genomic RNA. As you know, RNA itself is very unstable. Single-stranded RNA is even more unstable. So it's held in place by this protein called nucleocapsid. So if you look at this schematic of the virus, and if you look at an electron micrograph of the virus, which you can see that you can clearly see the viral envelope, and then you can see the club-like projections on the viral envelope. Now, these club-like projections is what makes the virus appear to have a crown, which are known as corona. That's hence the name of this family is called coronavirus. Now, what happens during the viral infection? So, the process of infection, of any viral infection, is a matter of probability. The correct virus has to meet the correct cell, which has the correct receptor, which is almost like a lock and key mechanism. You cannot have random virus infecting a random cell, which is why not every virus infects every person in every cell. So, you have the virus with the spike protein, and it scans around, it's in different it's in droplets, and if it comes in contact with a cell that has the ACE2 receptor, the spike protein initiates binding to the ACE2 receptor. ACE2 is angiotensin converting enzyme, which is distributed in the vascular endothelial cells, which is your ear, nose, throat, and lung. So predominantly the viral infection is a respiratory infection. And once the spike protein binds to the ACE2 receptor, a trigger takes place in this in the viral envelope and in the cellular receptor, where a membrane fusion takes place, and the viral genome is released into the cellular cytoplasm. And if we look at the overall life cycle, I'll take you through different steps. You have the viral genome unfolding, and the genome is released into the cytoplasm. If you're observant, you would have noticed that there is a 5' to 3' orientation to the genome, and this resembles a cellular mRNA. The entire virus life cycle takes place in the cytoplasm, and once the viral genome is released, the cell is tricked into thinking that this is a cellular mRNA, and the cellular translation mechanism starts translating this mRNA entirely. The first protein complex that it starts translating is the RNA-dependent RNA polymerase. This is very important for us to note, because in the general life cycle of a mammalian cell, you do not have an enzyme that is an RNA-dependent RNA polymerase. So, which makes it very interesting, because this is a very unique molecule that can be identified in the cell that has nothing to do with any host background contamination. So when you have the virus infection taking place, nowadays when we do the throat swab for the viral infection, the target for the real-time PCR is the RNA-dependent RNA polymerase. So, so this also makes it the real-time PCR test extremely specific and accurate. The RNA-dependent RNA polymerase further continues to make more copies of the genome, and it transcribes the smaller genes in the viral genome as well to make the other proteins. Now, this is what the genome looks like in a schematic. Like I said, it's a single-stranded positive sense 30 kb RNA genome. It has a 5' cap and it has a 3' poly A tail. So this 5' cap and 3' poly A tail does a great job of mimicking the cellular mRNA and hijacks the cellular translation mechanism. Now, the 30 kb RNA is important to note because coronaviruses have the largest of all known RNA viruses, are the largest of all known RNA viruses. Now, most of your RNA viruses are extremely error prone and prone to a lot more mutations, recombinations and things like that. However, coronaviruses generally have a pretty stable genome, and I will take you through the functions of the different proteins in its genome. So the first 2/3 of the genome, this is not represented to scale here, this is actually the first 2/3 of the genome, which makes the RNA-dependent RNA polymerase. And it's one of the few viruses that have a proofreading activity. If you know, viruses only carry the least amount of weight that they need to carry. Proofreading is generally not one of them. However, with a 30 kb genome, the virus doesn't have a choice but to carry a proofreading activity as well. And the virus, the ORF1A, 1B proteins, also, or open reading frame 1A, 1B proteins, are also known to suppress cellular immunity. As you know, there are different ways in which a viral infection is recognized by the body. One of the most common ones is the presence of double-stranded RNA. We do not have double-stranded RNA in the cell. So when these double-stranded RNA molecules are found, RIG-I, which is a cellular surveillance molecule, comes across it, it immediately activates the interferon pathway, which will shut down the protein translation in the cell. However, when these proteins bind the double-stranded RNA quite tightly, RIG-I will not be able to detect the presence of double-stranded RNA, and hence the cellular immunity is suppressed. The next is the spike protein, which is a large protein. It has the receptor binding domain and it initiates the infection. Like I said, it is a large club-like protein found on the viral envelope. Then you have ORF3, which plays a role in virus release. We have the E protein, envelope protein, which is a small molecule which actually plays a role in assembly of the viral envelope. When we do in vitro studies, we actually only need to express the M and the E protein to form the entire viral envelope. We do not need any of the other proteins. But in the viral envelope, the E protein is found in very small numbers, and it was the focus of my dissertation. This protein, we know, is there in a large quantity, but it is not really incorporated a lot in the viral envelope. So it plays a role in virus assembly. The M protein, which forms the backbone of the viral envelope, interacts with both the E protein and the N protein. The E and the N protein, nucleocapsid protein, is the one that binds the RNA. So the M protein binds the N protein and pulls the RNA in as the virus is getting assembled in the cell. The other ORFs, 6 to 9, they are known to suppress cellular immunity in various mechanisms and play a role in virus release. We also don't know some of their functions, and it is being studied. The nucleocapsid protein, the N protein, like I said, is required to stably bind to the genomic RNA, especially when it assembles into the viral envelope. We also know that it plays a very significant role in interferon resistance. A lot of viruses have at least one protein that reduces the effect of interferon on the viral replication. When an infected cell is targeted by interferon, which is, if you're in the medical field, you will know that for a lot of hepatitis infections, one of the treatments is actually interferon. But if you have an interferon-resistant molecule in the virus, the interferon treatment is not going to be as effective. Forget about the interferon applied outside clinically, but the interferon produced by the cells itself, which gives the virus a lot more time to survive, assemble, replicate, and go and further infect other cells. So when the virus, we went through all the proteins. So what happens is, all the proteins, the RNA-dependent RNA polymerase makes the mRNA for the smaller protein, and then they assemble in the cellular exocytic pathway in the rough ER, where they go through all your post-translational modifications. The membrane protein and the spike protein are going to be extensively glycosylated, and then they assemble in the ER Golgi intermediate compartment. It's called ERGIC. Sorry, I should have labeled that, but I have not. And then they are sent out of the cell by the regular cellular exocytic pathway. So what happens here? So this is what actually a transmission electron micrograph of an infected cell looks like. So these are the vesicles. So this is a very late stage infection, so you can't recognize other structures. Here you can see various stages of virus assembly. You have the budding that is taking place into these vesicles. Here you can see it very clearly. Then you have fully assembled virions that are being taken outside the cell, and these virions are present on the cell surface, which will eventually infect neighboring cells. And then you have these that have finished budding but just haven't pinched off into the vesicles. So this clearly shows what the virus like, how the virus assembles and leaves the cell. To understand this better, we were able to come up with a mechanism where the E protein, the envelope protein, actually causes a bend in the viral membrane, because there is no other reason for the membrane to physically bend to incorporate the viral protein. So you have the spike protein, which is the club-like structure. You have the M protein, which is represented in green here, which forms the viral envelope. You have the E protein on either side on the envelope, but not really getting incorporated too much in the virus. And you have the nucleocapsid protein, which are these beige dots that are bound to the genome. And this gets pulled into forming these kinds of buds that are getting assembled into virions, which are then transported out of the cell. It's important to remember that the entire life cycle of the virus is in the cytoplasm. There is no nuclear involvement as far as we know, even though some non-structural proteins are known to relocate into the nucleus. We don't know why it does that. So now, when we look at the entire life cycle, you have the virus that binds to the cellular receptor, you have the uncoating of the single-stranded positive sense genome, then you have the protein translation by the replication and translation by the RNA-dependent RNA polymerase, transcription and then translation of the various smaller proteins, and then you have the post-translational modification, virus assembly, transport out of the cell and infecting the neighboring cell. So, what are the various targets that we are looking at in terms of controlling the viral infection? All the vaccines are actually targeting the spike protein, the receptor binding domain of the spike protein, to prevent the viral infection initiation itself. Like we all know, prevention is better than cure. We want to stop the viral infection even before it happens. So all the vaccines that are being targeted and developed are being used to target this particular stage, where once the spike protein is exposed to the human body, antibodies can be triggered to prevent the binding of this to stop any further infection. The next step is for various molecules such as remdesivir, which is a very popular molecule, like you've all heard in the news. It actually targets this step, the translation and replication step, by binding to the RNA-dependent RNA polymerase, not necessarily the replication part of it, but the proofreading aspect of it, which means if the virus does not have a proofreading activity, it will be having a lot more errors being made, and that will reduce the overall viral numbers inside the cell. So at this point, so if we can target it in this step, which would be ideal. Right now we are trying to work both at this step for prevention and this step for treatment. So far, we cannot target the exocytic pathway simply because it is taking over the cellular pathway itself, and blocking this will affect the normal cellular function, but much more serious side effects than blocking any of these steps. So with this, I hope you've all had a brief understanding of how the virus life cycle is. And with this, I would like to finish my talk. At this point, I am a diagnostic lab and I run a lot of molecular diagnostics and test development, and this is what I do. And at this point, I will happily take your question.
Thank you very much, Madam, for the informative and interesting talk. Now I request all the participants to send their questions through the chat box, and the expert will be clarifying your doubt. I request Dr. Sudharani to moderate this question answer session.
Thank you, Rukmini. Yes, Dr. Pavithra, it was a very wonderful session with you are giving insights into the molecular biology of the life cycle of viruses. And some participants have raised their questions in the chat box. There is one Ms. Soumya who wants to know why is it called as novel coronavirus?
At this point, it's no longer called novel coronavirus. It's called SARS coronavirus 2. Before the molecular characterization, it needed to be identified as a different coronavirus from the ones that were already circulating. That's why at that point, it was called a novel coronavirus.
Thank you, ma'am. And there is another question from the same candidate, Soumya. What is the marker gene for phylogenetic analysis of COVID-2?
So they have been doing that using the spike protein.
Using the spike protein. Yes.
Using the spike protein.
Okay. And the same candidate is asking one more question. How ORF1 suppresses cellular immunity?
Like I said, it does so by preventing the cellular surveillance molecules like the toll-like receptors, the radar molecules, from being able to see the double-stranded RNA.
Thank you, ma'am. And another question from Sivabalan, and he's asking like, does the coronavirus has lysogenic kind of cycle, life cycle?
Okay. So this is a very good question. A lot of people keep asking, will the virus get reactivated or not, which is typically what a lysogenic virus does. No, there is, as far as we know, coronaviruses do not have an inert or an inactive part of the life cycle. It goes all out, it kills the cell and gets out. It's not like your herpes simplex, it's not like your retroviruses where it sits quietly in the cells for years and then it reactivates. So that there is no documented evidence of that. But this virus is throwing so many surprises at us. We'll wait and see. As of today, there is no evidence for that.
Okay, thank you, ma'am. Thank you, ma'am. And there is one Rochana Pramnik who's asking, how does the change in the conformation of the hidden RBD of S protein helps in viral attachment and immune cell evasion?
This is going to be a big answer. Actually, we can talk a little more privately because this might be too structural oriented for a lot of people, and I'm not a great expert at this. Generally, whenever you have any two molecules binding, you have the structural conformation change that causes the membrane fusion to take place. Now, when you have the viral attachment, we still don't know what causes the membrane fusion, but we do know that it is the receptor binding domain that plays a role in this aspect of it. Now, a related answer to this question is, one of the reasons this virus is spreading so vigorously among everybody is the binding strength, binding affinity of the receptor binding domain to the ACE2 molecule is actually quite high, which means only fewer number of viral molecules are needed to bind to the cell to initiate the infection. So even if you are exposed to a small number of viral particles, I still don't know how many number of viral particles you need to be exposed to. Even if you are exposed to a small number of viral particles, your chances of getting the infection is quite high.
Thank you, ma'am. And there is one Siba Patnaik who's asking, is coronavirus translation is different from?
Yes. The rhinovirus is a completely different family of virus. So rhinovirus is what? Negative stranded RNA. So you have a completely different enzyme. You don't have the this type of RNA dependent RNA polymerase.
And there is one more question from Abhinaya. If the person dies of COVID infection, does the virus also die or it continues or exists like in the dead body?
Okay, so like I said, viruses are obligate intracellular parasites. The virus is entirely dependent on, not just this, this is entirely dependent on the functioning of the for it to continue functioning. Now, I'm not going to say that as soon as the person dies, the virus also dies. There might be a small lag time where the cellular function will slowly drop down in its activity, which will prevent the virus from further getting out of the body and infecting others. So is this answer clear?
Yes, ma'am. Thank you. And there's one more interesting question. Why a patient who is declared positive and again found negative and again positive all in a span of 1 week?
Okay, this question is, the answer is, I don't know. So we don't know. There is a longer, the answer is I don't know. There are many theories for answers. First thing is, was the initial and the final test real-time PCR? Those are the things that you need to look at. Real-time PCR is the most sensitive and accurate way of identifying the viral infection. So if these tests were not done, that's one reason to conclude that there is a discrepancy between the two. The second aspect is, a lot of people are saying that there may be leftover virions that are testing positive. I don't know what that means because RNA-dependent RNA polymerase only looks at an actively replicating virion. So I don't know what virion particles are being left behind, so I cannot answer that conclusively. But since you say 1 week, these are the answers. But we also know that for any coronavirus, the immune response is not very robust. For example, if you get a chickenpox or a mumps vaccine or even tetanus, you have an immunity from anywhere between 5 to 10 years to up to 50 years. For coronavirus, we keep getting colds back, so the immunity is not very robust. And so the antibodies are no longer there. So if we give it maybe a little bit longer time, there is a possibility of reinfection. So that may be the reason they are testing positive again. But in the span of a week, I don't know. Maybe technician error, sample contamination. Without laying blame on anybody, it could be any of these things.
Okay. Thank you very much, ma'am. Rukmini, do we have time for more questions?
If madam can answer, like it depends upon madam's time.
I have time till 12 noon, so I can answer all the questions that you
Thank you.
I think that there's a lot of questions. Two more.
Yeah. Please, by all means, I'm available till 12. We have another 30 minutes. If that's okay with you, I can keep answering the questions.
Thank you, madam. There is one more question. Can virus become airborne?
So airborne standards of infection is much higher than droplet transmission. We are considering, so the latest statement by WHO is that we are looking at the publication and we're wondering whether it is airborne or not. Now, is this question relevant to most of us? If we are not working in a hospital ward, it is not relevant to any of us because the way to prevent the infection remains the same. Cover your nose and mouth, avoid crowds. These are the two things you can do to prevent the infection. So without thinking too much about this, let's follow the same safety measures that have been established.
Thank you. And there is another question from Suchi Patakari. By studying, Anisha Gupta is asking, by studying the effect of this virus on bats, can we overcome the ill effects of this virus? Understanding that way.
See, that's a very good question, and animal models do play a huge role in our understanding of various biomedical problems that we have. However, we don't know why an infection in bats is not so severe. So will it be the best model to study? Maybe, maybe not. Right now, the vaccine trials are being done on macaques, and right now, healthy volunteers are getting actively infected with the virus itself if they have been through whatever screening process that's there. So bats may not be the best model to study the infection.
Are there any other animals, other than human beings, there are other animals are affected?
There was some stories about cats being infected, some stories about dogs being infected. Whether it's a dead-end infection or whether it will come back, we don't know.
Thank you, ma'am. And there is one more question on the structural aspects of the virus. Can you please explain on the what virus release mechanism?
Sorry, I didn't hear the question properly.
They are asking about the OR3 mechanism.
So OR3 also has been known, OR3 has been known to play a role, especially in SARS coronavirus, not in the model virus that we generally study is infectious bronchitis virus and beta coronavirus is murine hepatitis virus. Murine hepatitis virus does not have ORF3A protein, which we think actually functions evolved in parallel to the membrane protein. They probably had two copies made of it, and one continued to evolve and while one continued to remain stable. So in SARS, we know that it plays a role in forming the viral envelope, not SARS coronavirus 2. In SARS, we know that it plays a role in forming the viral envelope. So yeah.
Okay. Thank you, ma'am. And there is one more question. Case he is again asking to describe the functions of all non-structural proteins.
There are many publications you can look at.
Thank you. Again, question it is whether it is or natural. Whether it is a natural one, it is man-made.
This question has been answered to death. It is an absolutely natural virus. And just because we are knowing so much more about it doesn't mean it's artificial. We just technologically advanced enough to identify how quickly it came and where it came from.
Thank you. And another question is, how precise is the target of RNA-dependent RNA polymerase for RT-PCR?
How precise meaning?
How precise is the target of RNA-dependent RNA polymerase?
There is no RNA-dependent RNA polymerase in the cell. It's a unique viral sequence when you look at amplifying the genome. So that makes it extremely valuable target for real-time PCR probes.
Because can a positive RT-PCR indicate the presence of any other virus other than corona?
People that develop the primers are very smart, but they will not develop it for any other viruses. When you have all this genome information, what you do is initially you identify the unique sequences and develop the primer and probe accordingly. So that's what makes the test very specific. That's what makes the diagnosis so accurate. People that make it also know what they're doing.
Thank you for clarifying the doubts. And which cells are found in COVID-19 patients for long-term immunity? He's just asking for long-term immunity.
We don't know if there is even a long-term immunity or not. So we have to wait. People ask as though the virus is so well studied. We have to remember that COVID-19 was only discovered in December. In 7 months, the fact that we know so much is something amazing and incredible.
Okay, ma'am. Again, some questions are repeated. It has become possible. Yeah. We can.
Whatever questions you ask, I'll answer, ma'am. Because you're seeing more of the questions. I'm only seeing a few.
Yes. How the medical community confirms that the patient is recovered, not going to infect others? How they confirm the patient who is recovered is not going to infect others? So
these standards, I'm not a medical doctor, so I cannot answer this with absolute clarity, but the standard is getting a negative real-time PCR report. A negative real-time PCR report should technically indicate the absence of any replicating virions, which is at which point you no longer have the replicating virions and your infection is considered concluded.
Okay. Another question on the immunity. If the virus inhibits the cellular immunity, how the patients are recovered? Which mechanism?
The body is also clever. It's always a balance between the virus being clever versus the body fighting. It is a balance that always happens. Plus, now we have excellent medical care where we give a hands up to the body's defense mechanism.
Thank you, ma'am. And Another question, whether the spike protein is specific to SARS virus.
Spike protein is specific to coronavirus.
Coronavirus only.
Yeah. The whole family. The spike is just the receptor binding protein. All viruses have a protein that initiates infection. This particular one is called spike, and it's not identical to all the coronaviruses either. They are slightly different among different coronaviruses.
What are the cells that are having AC?
AS2 distribution is anywhere between the vascular endothelial cells to your throat, to your lungs, alveolar. You have small intestine. So there is a large distribution, which also explains the variety of symptoms that people are having to this infection.
There are on the conformation of protein changing. By changing the conformation of a spike protein can be the binding of the virus.
Theoretically, yes. I don't know how you will administer this particular protein, though. So I don't know. Theoretically, yes. But who would get it? There are a lot of different technical issues that I am not an expert to answer.
COVID has no symptoms in some people, doesn't show any symptoms, but the virus doesn't get activated to infect others.
See, there is nothing called an activated virus or an inactivated virus. We have to remember that, especially in the coronavirus lifestyle. Yes, a person not showing symptoms does not mean they are not infected. It just means their immune system is strong enough to suppress the side effects of the viral infection. Virus is still replicating, but immune system is keeping it under some control. You don't fall sick. However, when the virus is still there and it's still being spread through your aerosol droplets. So you can't assume anybody is negative. According to me, everybody is COVID positive. I am COVID negative, so I can keep myself safe. That is how I deal with my patients.
Asymptomatic carriers.
Yes.
So, ma'am, can we just take one or two more questions?
Yes,
and let the participants can ask the question because she has already given the number and she can just.
That will be great. We have 120 questions. I don't think we'll be done.
I know. Okay. Two more questions we can just take.
You can choose the questions and I'll answer them. For everybody else, please message your questions. You can email it to my address, or you can send it to me my WhatsApp. I will not answer calls a lot, but I will reply to every WhatsApp.
Is the present one is a mutated virus?
What does it mean? We are all mutations of different things, no? It is something that is, mutation is a genomic change, so we are all mutations of something. So I don't know what they want.
The symptoms are all common questions.
Yeah.
One question now. Can we use my RNA silencing method for blocking this virus spreading?
So how will you, so this is a question that I've been asked from the beginning. Theoretically, yes. How will you administer it? How will you ensure that it enters the specific cells and binds the RNA? Theoretically, yes, absolutely. But the next caveat that you have to look at is the virus also is able to prevent the detection of double-stranded RNA. When you use siRNA, you have the double-stranded RNA, which also plays a role in the immunity detection and the viral cellular response to the viral infection. So how will you do that? So the practical aspects of it, I cannot answer.
Thank you for.
Thank you so much, everyone. Thank you for asking all these questions. I know there are a lot of questions, but what I will do is I request you all to message me and then I will be able to answer your questions. Don't ask me if it is a mutated virus or if it is a bio-war. I will block your number. But other questions I will happily answer. Thank you so much. It's an honor being a part of Pondicherry University's Covinar.
Thank you very much.
Yeah. Thank you very much, Dr. Sudharani and Dr. Pavithra, ma'am. It was a wonderful question answer session. Now I would like to know whether our HOD, Professor P.P. Mathur, would like to ask anything or share anything here. Professor?
Yeah, no, thank you so much. I think it was a wonderful presentation.
Thank you, Professor Mantha.
And I think that we must interact with her for future discourses also and different. And it was a really very restrictive and very simple presentation which every student or anybody can understand. Thank you so much for listening. And whenever Corona is over, we would like very much to visit us in Pondicherry University.
Absolutely. It's been my pleasure.
Okay. Thank you so much. Thank you.
Thank you, sir.
Thank you very much, all of you. Now I request Dr. C. to propose the vote of thanks.
Thank you. Thank you, Dr. Rukmani. Dear participants, on behalf of the organizing committee of Covinar from the Department of Biochemistry and Molecular Biology, Pondicherry University, I express my sincere thanks to our chief guest, Honorable Vice Chancellor, Professor Gurmeet Singh, for taking his precious